This is Biochem Bits, and I'll be talking about chemistry in bits. I'll be speaking to the scientific methods and experimental techniques that are involved in biochemistry in this video, and I expect that at the end of this video, you should be able to evaluate the strength and limitation of some of these identified experimental techniques. So I'll start with defining what scientific method is as a background. Scientific method, I have simply put, as a systematic approach to investigating biological process or events in a bid to understand or acquire new knowledge. And just like every other science subject, it involves observation, in this case, live processes, formulating hypotheses, and also conducting the experiments. And in conducting experiments, the method that is used is very critical. The method involved is important in biochemistry because it allows reproducibility of results. So, following this standardized method, other scientists all over the world are able to replicate my experiment or whoever has carried out the experiments and verify what the finding really is. So, if there is any disparity of variation, it will be based on some other factors like environmental factors or uh, um, any other thing, but not the method itself. And this will help to minimize any form of bias that may want to arise in or in questioning of the results or the research output, facilitating the discovery of new or novel mechanisms and also improving upon pre-existing ones. And so finally it allows the systematic study of complex biochemical processes with confidence, because scientists all over the world are able to build a kind of framework for breaking down some of these techniques and managing the research questions that may arise during the course of research. In biochemistry, there are high throughput technologies, techniques that are involved in carrying out experiments, but I'll just speak to these five basic ones: spectrophotometry, chromatography, electrophoresis, polymerase chain reaction, and Western blotting. Now, I'll start by focusing on spectrophotometry, which is a quantitative analytical method that measures the intensity of light that is absorbed by your absorbing species or transmitted by it, and this is a function of the wavelength at which the absorbing species is taking up light. Now, the basic principle is the Beer-Lambert's law, which says absorbance is directly proportional to the concentration of the absorbing species relative to the path length and also um, the wavelength. The light that is coming through the spectrophotometer can be in different region. It can be in the UV region, UV visible region, the infrared region, as well as the visible region itself. The component of every spectrophotometer starts from the light source, which is sending out the light, a monochromator that would select the band or the region of the light that is coming in, a sample holder where you have already put your sample that contains the absorbing species, and a detector system that will read how much light was transmitted, how much has been taken up or absorbed, and then we have the recorder on the other end. So, application of spectrophotometry in biochemistry is very wide. One is that we use it in enzyme kinetics and also DNA RNA quantification, cell growth measurement, and protein concentration determination. The most common one is the determination of protein concentration and enzyme activity. This technique has its own limitation, and it is important that we note it. It only works for light absorbing molecules and can be affected by interfering substances. So we have to keep in mind some of these pros and cons in designing the experiment and knowing which one will be better for us. So if your sample does not have an absorbing species, please don't go use spectrophotometer. There are other methods that will be able to also quantify and identify bioactive components. It's also advantageous in that it is fast and it is highly sensitive and also quite accurate. The second technique I'll be talking about is chromatography, and this is also a very versatile separation technique. What it basically does is that it partitions the components of the mixture between the mobile and the stationary phase. The basic principle is that components of the mixture move at varying rates, and this varying rate is dependent on the size or the shape, which is between the mobile and the stationary phase. The stationary phase can be in the form of liquid or gas, liquid like methanol, ethanol, and the likes. But while the stationary phase can be in the form of gel or beads, while the mobile phase can be in the form of liquid or gas. We have methanol, ethanol, and also in the form of gas. In a system like high performance liquid chromatography, there is also an absolute need for a sample injection system and detector system. There are different forms of chromatography ranging from liquid chromatography to gas chromatography to thin layer chromatography, and even the very one that we're familiar with, the paper chromatography and size exclusion chromatography. Based on the advancements in science, the most common one now is high performance liquid chromatography and gas chromatography. And chromatography also has this advantage because it can be coupled to other systems like LCMS, liquid chromatography coupled mass spectrophotometry, which will help us to identify or even get the mass of the identified species. One of the important advantage of chromatography is that it carries out both quantitative and qualitative analysis. So if you're looking to analyze both quantitative and qualitative analysis in your experiment, I think it will be better off to pick chromatography because it will give you both findings. And in protein purification, chromatography has been applied. It has also been applied in drug discovery and development, where you're able to identify the active components or active ingredients in a particular sample and then be able to narrow it down and separate some complex biological components. But some of the disadvantage or limitation to this technique is that it takes more time and can be quite expensive. By starting with movement of electric introduction of electric field that is applied across the gel, and smaller molecules will definitely move faster because they are lighter, and larger ones will move more slowly. So molecules with more electric charge, we have the polar and non-polar. If you have those some I'm giving that as an example now. The polar molecules will move faster while the non-polar will retain more, and after some time the molecules will separate into two different bands or different bands, and this you can now bring out for visualization or for further experimentation. The different types of electrophoresis, which moves from agarose gel electrophoresis, SDS page. These are the ones that are quite common now. And we also have the 2D electrophoresis. I'm not very familiar with 2D electrophoresis, but I've heard of and know of isoelectric focusing and the likes. So the rate of migration in electrophoresis basically is dependent on the molecule's property and the medium that you are using. The medium has to do with the charge that you've been able to that you've introduced to the system in the electrophoretic chamber, as well as the property of the molecule itself. So if you know the property of your molecule, you will be able to separate them based on that characteristics, and it has been applied most importantly in DNA fragmentation and also in protein separation, also purifying or assessing biological um, samples. And I think I've also seen it being used in forensic profiling. This is also very important. The limitation of electrophoresis, lest I forget, is that there is need for charged molecule. As I mentioned, the molecule has to be charged. Some of them has to be charged, and so that this is what will initiate the separation. And this charge, this electrical charge that is introduced, sometimes could polymerase, which serve as the enzyme. For example, the Taq polymerase will do the extension after the primer, forward or reverse primer, has been able to identify the sequence from the template DNA that will be copied. We have the NTPs that will be added up to. Those are like the nucleotide that are added up, and the buffer solution in the system that will maintain a viable system or a stable system for the DNA polymerase to have maximum activity. The thermocycler basically is to amplify the the target sequence, and you have millions of copy of what you really want. Now, the advantages of PCR is numerous, and that is why it's one of the go-to technique in biochemistry. It's highly sensitive and also very specific, even though there can there may be room for error if you're not careful with your experimentation or your procedure. So it requires more meticulosity, and you need to pay more attention to it. So it requires minimal material. That's also very important. You don't need to take so much material to get what you really want. You require just minimal material. It will amplify by itself and make what you really want to achieve, to help you to achieve what you really want. For example, in gene expression analysis, genetic disease diagnostics like identifying single nucleotide polymorphism, mRNA expression level, your forensic DNA analysis like identifying who or gene mapping, cloning and genetic engineering, and evolutionary biology studies. There are variation in PCR. It could be quantified like qPCR. We also have reverse transcription PCR, RT-qPCR, and also multiplex. These different ones has their specific type of experimentation that you can use them for. So if you really want to use them for research, then you will go into the nitty-gritty of what each of this type of PCR involve. Basically, what it does is to amplify your specific target, and it is limited in the sense that it requires a target DNA sequence for primer design. So if you don't get your primer right, there is a high tendency That's what you end up amplifying is not your area of interest. The last that we're talking about in this video is Western blotting, and has to do with detecting and quantifying specific proteins amidst other protein. And in this case, you have to be more specific. That is why Western blotting combines both the separation technique as well as identification technique. The separation technique has to do with the gel electrophoresis, where you have a specific antibody. You have a specific separation from amidst the based on some features from amidst the mixture, and you now have an antibody that will go pick it up, and the detection system. Basically, I will just run through some of the process that's the key steps that are involved in this. First is the separation. Like as I mentioned, you have to go through the gel electrophoresis to separate some of those protein based on some of the characteristics like like size, shape, and also the charge. Then you transfer this identified bands or this gel based on their separation into a membrane. The membrane could be a nitrocellulose membrane or a polyethylene difluoride membrane. And then you block. This block is to prevent non-specific antibody from binding. The first thing you will introduce after blocking is the primary antibody. So the system is more like a sandwich system. The primary antibody will be picking up the specific protein that you have interest in, and that's why your primary antibody is defined for the protein, is to identify it and bind with it. And the secondary antibody will be able to identify your primary antibody, but in this case it's carrying a detector system. So when your secondary antibody binds with the primary antibody that has already bound with the proteins, look at it like this. You want to make a sandwich, you have the components, the first background, like the bread, the loaf on the down part, and you have your beef, and you have some vegetables put on it, and you now have the upper one to cover it. So it looks somewhat like that. It's more like a sandwich system. The detection system is what will quantify how much of this substrate can be detected. Often you use the chemiluminescence procedure system to identify it. This system is also very powerful, and it analyzes a protein separation. It's also very, very sensitive, and it can detect as much very small quantity of protein. As I mentioned, one very important thing we use Western blotting for is post-translational modification study and protein-protein interaction. Data that is collected from Western blotting is used to provide some semi-quantitative data on protein presence and protein abundance. So no matter how small or minute a protein is, it's able to provide information about it. Okay, do we have more of it? And even the status of the protein. For example, I once worked on BDNF and was able to identify the pro-BDNF and also the matured BDNF. So the stage those proteins are, also you will be able to give a more specific information about it. So I think the limitation in the case of Western blotting is that there is need for optimum optimization for each protein of interest, meaning you need to be sure that the antibody that you're introducing is really to able to identify that thing, and the potential for non-specific antibody binding is possible, which will then give some false negative or false positive. So it also takes time. Times you have to incubate for some days and the like, so it takes more time. I'll speak to biochemistry data analysis. I want to say more categorically that data analysis in biochemistry is very important, and it is critical that we know how well we present our data, what type of analysis or statistical analysis it will go through, identifying the level of significance and the p-value to be able to really say if this had occurred by chance, or it's a significant process or a significant event relative to what the control is or the standard is. How well you present your data to it also matters. For example, you may be presenting it linking the network of metabolic pathways, talking about the protein-protein interaction in the 3D form, and also presenting it in the graphic or graph way, and you have box plots in different ways you feel you can express or explain your data to someone that wasn't in the lab with you, it's best to present it that way. And it's still a process in biochemistry that everyone, including me, is trying to measure up to and learn more and know more about how best to present my data. There are some very important things to consider in presenting some of this, some tools like GraphPad Present that you should familiarize yourself with, like R Studio, Python, and ImageJ. In ethical consideration in biochemistry, it is a very important topic because key consideration include the honesty in data, this proper citation, and responsible authorship. For example, for human subject research, you have to get informed consent, you have to get some privacy protection signed, and in animal research too, which are some of the things where I've had experience, you have to follow the 3R principles like replacement, reduction, and refinement. They don't joke with it when it comes to ethical consideration in animal research. And you also have to show that, okay, this research is very important. It's important that you use animal and you handle your animal appropriately following the standard protocol. Biosafety and biosecurity, don't leave some things out of the lab, don't bring some things into the lab, especially when it comes to handling some very hazardous materials like genetically modified organisms and the likes. So I will say that even though there are other ethical considerations like social responsibility, conflict of interest, and the like, there is absolute need for a well professional conduct when it comes to biochemical research, and this will help to maintain the integrity of research and the practice of your research. And so, to wrap up, I would say that we've been able to look at scientific method and some of the techniques that are involved and the ethical consideration, as I mentioned just now. I will advise that you get to know about the strength and limitation of some of these techniques and you know which best applies to the research that you want to carry out. And so please try to like this video if you do gain something from it. Share with people that you feel are just coming into biochemistry field and want to know more about the process, and also subscribe to the channel so when we have a new topic you can always get a notification about it. Thank you.